Good morning, everybody. My name is Rita Russo, and I'm from Cardiff University, and I will talk to you today about how we can use genetic information, specifically information downloaded from public databases in the management of African bovid species, and I will use the red hartebeest as an example. We have developed a management guidelines for other species as well, such as carnivores, but I will focus just on one ungulate species for today's presentation. I am presenting this work on behalf of my co-authors, as you can see here, and we are representing a number of organizations, universities. I'm based at Cardiff, but I do a lot of work in South Africa with colleagues in South Africa, but also Namibia. We have one colleague from Namibia, representing the Ministry of Environment and Tourism. And this work is really part of the Conservation Genetics Specialist Group, specifically the African node, of which I am one of the coordinators. The work that we've done thus far has been funded by the Global Challenges Research Fund. Okay. Some background information. So we all know that biodiversity consists of three layers: ecosystem diversity, species diversity, and then genetic diversity. But often genetic diversity is overlooked when it comes to policy. And this is really due because of the fact that people don't understand genetic diversity. There are often gaps. Think about species that have very, very large distributions. There's sometimes gaps in those distributions in terms of the genetic information that is available. It's difficult to also understand published results if you are not a geneticist. And the field of genetics has evolved over time, and it's therefore been refined over the years, so not always easy for people to understand. There's often incongruence between what you see with genetic data and what the taxonomic revision shows you with regards to a specific species. So it's very difficult to put the two together and come to sort of a consensus. There's also often a lack for the appreciation of the need or the obligation to really understand and conserve genetic diversity and genetic structure, but not just genetic diversity and structure, also the processes to allow for speciation to happen and also for the adaptation of species. And there is often no common objective or approach. If you think about different countries having the same species, such as in Southern Africa, they don't have the same approach or objectives, or it could even be within a country where you have provisional regions and they don't agree, or they have slightly different objectives or approaches. So because of this information as background information, we realized as a group that there was a real need for guidelines, genetic guidelines, to actually help develop clear and concise guidelines for the decision makers, specifically for the translocation of large mammals or mammals in general. So in South Africa, permits need to be issued for animals to be moved around, and often those permits are issued without taking any genetic information into account. So what we did then as a group, based on the best available science on the genetic structure and diversity for a number of species, we then developed some guidelines. And we also try to actually keep in mind or try and understand the underlying processes that actually led to the diversity that we see in species today. And we also kept in mind that often these guidelines that we develop based on the genetic information might be very different from what is published in terms of the taxonomy of species. So we're trying to put the two together. So the whole idea was to come up with pragmatic guidelines, but incorporating genetic information into those guidelines, taking into account the actual practicality of implementation. It's not always easy to implement guidelines or suggestions. So we had to take the practicality into account, and also taking into account the history and the movement of species in the past. And this then kind of led to concise but also consistent decisions in terms of the management of genetic diversity and structure for mammals in Africa. So the work that we've done thus far only focusing on Southern Africa, therefore we only have partners from South Africa and Namibia at the moment, but the idea would be is to kind of widen the project at a later stage and include other African countries as well. Okay, I'm going to quickly explain to you the process that we followed in short. So we first started off with some literature reviews. We identified some species of interest in terms of translocation, so species that get translocated quite often in Southern Africa. So those were 3 species that we initially started with, sable, hartebeest, and the African buffalo as our initial 3 species. And then we also downloaded all the information that we could find in terms of genetic information from publicly available databases. And then from there on, we've expanded it to 13 species, and currently we have 17 species. So then once we had all the literature, relevant literature for each of the species and the associated genetic data, whether it was sequencing data or microsatellite data, we then kind of reanalyzed some of these data sets, and we then kind of built these draft guidelines. And this is just kind of to show you what we have in the guidelines. References, the name for the species that should go on the permits, management levels, management boundaries, limitations and assumptions, and then actual area for priority research for the future. And then we also try to develop these maps indicating conservation units based on the genetic information to say these are the different conservation units, and within these conservation units there's also some smaller units. We generally refer to those smaller units as MUs, management units, and the larger units as ESUs, evolutionary significant units. And we don't generally mix animals between ESUs, but within MUs, the management units, we can move individuals. And these then draft guidelines were reviewed by taxon experts, geneticists, ecologists at national and also international level. And then we had 3 workshops within South Africa and Namibia, and we then kind of finalized our guidelines based on the outcomes from those workshops. Okay, so now I'm going to move on to the hartebeest, and I'll give you a quick example. So this is the distribution of hartebeest here, right in the top corner here of the slide. And you can see here, historically, we had 8 subspecies. One of these subspecies right here in the north has gone extinct. It's been declared extinct in 1994. And this is the Bucelophus subspecies. In 2001, Flagstad et al. Published a paper, and they used samples from across the range. And based on their information, which was based on mitochondrial DNA sequences, D-loop sequences, and cytochrome B sequences, they kind of identified a big split between the northern subspecies and the southern subspecies. And based on this information, we were then allowed to actually recommend some management units and ESUs for hartebeest as a whole. So in our current guidelines, we basically then said we have 2 ESUs. We have a northern ESU and we have a southern ESU. And then within this southern ESU, which is the ESU at the moment of interest, because the project will expand to the rest of Africa at a late stage, we actually have these 2 subspecies, the Kama subspecies in the sand brown color, and then the Liechtensteini subspecies in the more orangey color. And these 2 subspecies are basically the equivalent of 2 management units, okay? So information that we could download from GenBank already gave us some idea of where the genetic diversity is and how the genetic diversity should be split up or compartmentalized. This is basically still from the Flagstad et al. Paper, but what I want to really show you here is if you are doing a phylogenetic tree, you get very, very similar result. You get a southern lineage and you get a northern lineage. Within that southern lineage, you have the Kama and the Liechtensteini subspecies. Also, this is a network basically showing exactly the same information, but what I want to draw your attention to is that for Namibia, for the southern subspecies, we only had samples from Namibia and Botswana. So this is basically the guidelines. You can see all the different sections, and then we end up with these maps. And these maps are basically just showing the ESUs in the smaller map. So you have the northern ESU, which is in the blue, and then the southern, which is in the green. And then within that southern ESU, in these dotted lines, you can then see the one management unit here, which is southwestern, and then the other management unit, which is southeast central, which correspond to the two subspecies that we have. Okay, but there was basically a problem with that current interpretation of the published literature because we had no samples from South Africa, only samples from Namibia and from Botswana. And the samples we did have from elsewhere, they were based on very small sample sizes. So specific geographic regions and also specific subspecies. So we have gone and we've added subsequent information. So we've sequenced more individuals. So this is based on D-loop sequences. And basically what you can see here is all the South African individuals here up in the north, but also we have a South African haplotype here, CA14, that is very, very diverse from all the other South African haplotypes. It actually groups closer. LI is Liechtenstein, it would actually group closer with the other subspecies that we have in Southern Africa, as opposed to actually grouping with the Kama subspecies in South Africa. And then this is a major, this is another subspecies, the closest related subspecies from the northern group. So already from this, taking information from GenBank, so we downloaded the sequences from GenBank, putting it into the context of new information, we can already see that there's something really interesting going on here in South Africa, because you can see in this little map here at the bottom, the post-1999 records for hartebeest in South Africa and the pre-2000 in the sand color, they are also records for hartebeest. So we still have a lot of hartebeest in South Africa, but South African samples were not represented in the original data set. Okay, then in terms of conclusions, well, using the available sequence data from publicly available databases really help us to kind of define those broader scale patterns of genetic diversity within the hartebeest, red hartebeest here specifically. The finer scale patterns was not really always well resolved or understood, but adding new data with the existing data on databases really help us to kind of resolve those finer scale patterns. And this really led then to the description of ESUs and MUs that we can use in conservation management of species. So the next step would then be to expand this project to other African countries. So actually look at the whole hartebeest complex. So we now have samples from museums that we will add, and we are also busy generating microsatellite data, so a different marker that we will put together with already published microsatellite data for this species. And then our next step would also then be to move on from the mammals and actually start developing guidelines for other taxonomic groups such as reptiles, but still actually using information that is available on GenBank or DryHat, any other publicly available databases. So I hope this has illustrated to you guys how we can actually use sequencing information or microsatellite information, genome information that's already available on public databases, and how we can actually put that into a broader context, and we can use that in the end for the management of species. Thank you very much. And yes, I just want to say sorry for my voice, but I have a slight sore throat, and I'm happy to take any questions.